Today we'll talk about role of complement. This chapter deals with different components and pathways of complement. How do we regulate them, and especially what are the functions of anaphylatoxins, and how do you regulate complement activity in body? This particular table is basically giving you a function of complement proteins, and all of these proteins are important. And when we look at some of the disease states, we basically go and determine the levels of this complement protein. So we usually would like to know the complement levels of C1, C2, C4, especially when we are looking for immune complex diseases, especially when we look at type 2 and type 3 hypersensitivity reactions. C3 is an important opsonin. Basically, those people who are lacking this complement protein, they have a deficiency of this protein, and hence they are susceptible to capsulated bacteria, because you have to have opsonin capsulated opsonin looking at the capsule and help to phagocytose the bacteria. Likewise, C5 through C9 are important for susceptibility to Neisseria, and Neisseria, of course, is an important STD. And factor D and properdin factor P, if you are deficient in that, you will again have a susceptibility to capsulated bacteria and also Neisseria. But in this case, there is no immune complex disease. Factor I, basically the deficiency is similar to C3, so that's kind of an opsonin. And then we have DAF and CD59. When you have deficiencies of these two factors or complement proteins, you have an autoimmune-like condition, especially paroxysmal nocturnal hemoglobinuria. And finally, one more important complement protein is C1 INH, and that basically is an important cause of HANE, hereditary angioneurotic edema. Now, initially, when complement proteins were discovered, they found out that there are some proteins, especially in the serum, and they are heat sensitive. And that basically was considered as a complement to the ability of antibodies to kill bacteria. So they would go and bind to those bacteria and help antibodies to go and kill them. We right now have about like 30 different circulating membrane express proteins. And if you look at complement system as a whole, so you will see that it's a major system. It plays a major role against many infectious organisms. And basically, we consider that as a part of both innate and antibody-mediated acquired immune responses. And the other important thing for complement is, since they are proteins and they are synthesized in liver and by the cells which are involved in inflammatory response, so whenever there is a problem with the liver, like hepatitis or any other liver disease, you will have again deficiency of these complement proteins, and you will be prone to all those factors that we discussed earlier in the table. Since complement activation can lead to very possible effects which will cause inflammation, and which will be an important aspect in terms of hypersensitivity type 2 and 3 type reactions, hypersensitivity reactions. So we basically want to make sure that we have the complement activation tightly regulated. Now, if you look at the three different pathways for complement activation. You will see number one is called a classical pathway, and number two is called lectin pathway, and number three is alternative pathway. The most important thing that you need to remember is that how are these complement pathways activated. For example, for classical pathway, it is activated when C1 binds to an antigen antibody complex. So that's the most important aspect as far as classical pathway is concerned. Whereas in lectin pathway, this can only be activated when mannan binding lectin MBL. It binds to terminal mannose residues on the surface of either gram-positive or gram-negative bacteria, or even fungi or yeast. So that's most important thing. The third important thing is that in alternative pathway, the complement gets activated when component C3b is deposited on the surface of pathogens. So all you need to keep in mind that these complement proteins are not normally there in the serum as already pre-activated, so they need to be activated. So there has to be an inciting agent that will cause the activation of these particular proteins. Now, this basically is an important summary of whatever we've just discussed in terms of classical lectin and alternative pathway and how does complement activation takes place. So again, I would want you to pay attention to the key activators. What are the key activators? How do you initiate complement components? And what are the components which are common to all pathways? And then, of course, what are the major functions or activities generated by this, by either of these mechanisms? So again, you will see that classical pathway initiating complement component is C1. For lectin pathway is MBL. For alternative pathway C3b, and all of them basically converge on cleavage of C3. So if you're asked that what is it that all three pathways converge on, so that's C3. Now the other important thing in a brief manner, especially in this cartoon, will give you the components which are common to all pathway. So every protein, like for example C3, it basically would break down into an A and B component. So you'll have from C3, C3A, and C3B. Now C3B especially again is an opsonin, and is also important in terms of application loops. It will go and help alternative pathway. And then subsequent steps like C5 again will split into C5A into C5B, and then all the way through C6 and C9. So if you look at the last line over here, you'll see these are some of the major activities generated in pathways. Again, the secretion of cytokines, the secretion of anaphylatoxins, and then again, killing of pathogens. So these are the common things that we normally see in complement activations. So what I would want you to know, especially in this chart over here, is that how again complement pathways are activated, what is it that they converge upon, and then again since there are 30 different plasma proteins, so we just concentrate on the major one. So the most important one in this case is C3, and then when C3 is cleaved, it's going to cleave into C3A, which is basically anaphylatoxin, and that basically C3A would induce a very potent inflammatory response. Whereas C3B is again is an opsonin, and as we discussed earlier, it's going to deposit on the pathogen surface, and of course, when it is sugar coated, it's going to enhance pathogen uptake by phagocytic cells. Now, also keep in mind that again, as we discussed earlier, the last line of that cartoon, what is the result of complement activation? So this figure shows at least the three major components of complement activation. Number one, the production of opsonin on the pathogen surface. Number two, the synthesis of fluid phase molecules that enhance inflammatory response. And number three, direct killing of the pathogen, which we'll discuss in a while. So all these activities together will rapidly remove or directly destroy pathogens. So that's the defense mechanism. So they're part of the defense mechanism. Now, a little bit of detail. Let's take each one of these pathways separately and go for some of the important components that they are there. Number one, classical pathway. Again, there are different component proteins, C1 through C9, and basically these numbers are in order in which they were discovered. And all different proteins components, they basically break into an A and B. So you have this lowercase letters called A, and basically most of the time A we say anaphylatoxin, and B basically is an opsonin. And if you look at figure 13.2, in the classical pathway, as I said earlier, well, the activation factor is C1, and C1 binds to Fc region of the antibody. And these antibodies, especially two, are IgG or IgM. So IgG and IgM are basically complement fixing antibodies, and this was the question that we asked before as well. And also keep in mind that IgG, IgM, especially IgG1 and 3, they are very effective activators of the classical pathway. Now, if you again look at the early steps in activation of classical lectin and alternate components pathway, you'll again see that they all lead to formation of C3 convertase, which is an enzyme, and that will cause the breakdown of all the different steps which are there. And you can see from here, we don't want to go in detail, but it will give you an idea that for each and every step, there is an A portion component, the B portion. A is going to help in causing inflammatory response. It's going to attract more cells of inflammatory response, whereas B basically will go and label those bacteria and then will make them palatable or eatable for the phagocytes. So you will see that a typical pattern. I don't want you to remember each and every detail, but just important aspects of the important classical pathway. And especially in this figure, I would want you to know that most of them will lead to C3 convertase, which is an enzyme. If you look at the lectin pathway, again, important question is how is lectin pathway activated? One thing you have to keep in mind is there's a terminal mannose residues. These are expressed both by gram-positive and gram-negative bacteria, also fungi and yeast. And these are activated in the absence of antibodies. So it's a part of innate immune defense. And if you look at the early step, you will see initially when a pathogen that expresses mannose and binds to mannose binding lectin, and once it binds to that, and then there's a sequence of events that happen all the way from C4 to C3, and then again C4B2A on the surface of bacterium. And again, if you look at the step over here, you will see all those steps which are there in terms of comparison of all these classical lectin and alternative pathway. Now, let's talk about alternative pathway again. As usual, we need to know how this pathway is activated. Very important question for each and every pathway, and it's going to come over and over again. Now, you have to remember that the first two pathways are initiated in the presence of a specific antibody, whereas this pathway is activated by almost any foreign substance in the absence of specific antibody. So that's the most important differentiating point. It's also an effector portion of the innate immune response, and the early steps again are deposition of C3p, which is an opsonin, on the cell surface that will initiate the alternative pathway. Again, the same figure is there, and you will see the sequence of events pretty much the same. Now, if you look at the steps which are shared by all pathways, activation of C3, C5, there is the C3 cleavage, and that cleavage takes place when there is an enzyme called C3 convertase, and this convertase will cleave it into C3A and C3B, as we discussed earlier. C3A is released into fluid phase, whereas C3B continues the complement activation cascade. Also keep in mind that C3 convertase also allows the next component in sequence, which is C5, and it will bind and cleave. And again, C3 convertase with C3b will lead to C5 convertase, and then again C5 will break down into C5a, which is in the fluid phase, and C5b will bind to cell surface. So this is a kind of a common shared pathways for all pathways, that they will come on C3 cleavage, and then C3 convertase will come and split them up into C3a and C3b. As we discussed earlier, C3b is also important in terms of amplification loop. Now, in this specific comparison, you will see that the again cleavage of C3 by C3 convertase, and then C5 by C5 convertase in the classical and lectin pathway, which is like top panel, and then the alternate pathway, which is a lower pathway. Again, you need to understand in all the pathways, C3 is cleaved into C3b, which deposits on the cell surface, and C3a is released in the fluid. Similarly, C5 is cleaved into C5b, which is deposited on the cell surface. C5a is released in the fluid phase, and in alternative pathway, the stabilization of C3b by a properdin increases C3b deposition on cell surface. So that's basically an amplification route that we discussed earlier in terms of complement activation. Now, if you look at the terminal pathway, lastly, terminal components are C5b to C5, 6, 7, 8, and 9, and they're very important because they're going to form a very important membrane attack complex that is called MAC, that will result in cell lysis. So that's very interesting, that I'm going to discuss in a while. So if you look at this picture, you have all those C5 through C9, and they form a MAC, membrane attack complex. So they kind of come together. Initially they are separated, when they form complex on the cell surface. So they will kind of bore a hole, a pore into the cell membrane and disrupt the cell membrane, and you have this osmotic disequilibrium. So you have all the salts and water, moving in and out of cell, and will totally disrupt the cell membrane. So the whole idea is that multiple C9 components, they bind to this complex and basically polymerize. So this polymerization of that will lead to create a channel, and that will disrupt the osmotic balance of water and ions, and then you will lose the equilibrium there. Now, as I said earlier, you need to know how this important component, especially if a component is regulated. Keep in mind that they are found on the surface of many mammalian cells, and there are a bunch of receptors for there. Some of them are found in fluid phase, like factors H, I, C1 inhibitors, C4b binding proteins. So these are some of the regulators that we have in the system. Now, if you pay attention to the most important one that we had in C1, how do you regulate the complement activity? There is C1 esterase inhibitor. So there are some inhibitors, C1-INH, which is a serum protein, and this inhibits the first step in the activation of classical complement pathway. Likewise, we have C4b binding protein, which is found in serum and decaying accelerating factor, DAF, and CD55, and then again complement receptor 1, CD35, membrane cofactor, MCP, membrane cofactor, which is also called CD46. They are all widely distributed cell surfaces that regulate classical and lectin pathway C3 convertase, which is like a common joint where all these pathways will come. Now, the other important regulators that we have are factor I, which is again the serum protein, and the function of factor I is cleave C4b on the cell surface after C2a has been displaced. This particular factor requires presence of one or more cofactors, and these are factor 1 mediated cleavage and a bunch of other receptors. Factor H again is a serum protein which stops alternative pathway if binds with C3b. Very important concept, and you have to memorize it, that factor H is a serum protein, stops alternative pathway if bind with C3b. It can also bind to C3 convertase in the alternative pathway, and it will displace the B portion for that, and then again it will stop the further activation of complement cascade, because this is going to go in a sequence of event when you go from C1 through C9. So if you can block that, you block all the activity for that. Again, you can see, without going into detail, These are regulators of C3 convertase, especially in classical pathway A, alternate pathway B. The regulators may dissociate the convertase and cleave the complement component on the cell surface or act as a cofactor for the cleavage. So these are some of the regulatory proteins in classical factors that you can see from here, factor 1, factor 1 in both cases, and some of the dissociation can happen between a sequence of events from 1C1 through C9. So you can stop that happening. All those cleavage of A and B can be stopped by these particular cofactors that are required for cleavage. For example, if there's a C4 binding protein that exclusively regulates the classical pathway and factor H that regulates the alternate pathway, and then bunch of factors that we discussed earlier, factor I, DAF, CR1, MCP, they regulate both. So these are some of the important functions for that, because these complement proteins are not running around in serum already activated form. So we have a lot of control mechanism, and we want to make sure that they don't go out of control. Otherwise, there will be such an inflammation that will be out of control. Now, you'll ask yourself, well, why do you have so many complement proteins? Why do we care? Now, we do care because, of course, for capsulated bacteria, you have to have opsonin. For some of the functions of antibody-mediated functions, antigen-antibody complexes, we have to have opsonization, especially IgG and IgM, as we discussed earlier, IgG1 and IgG3. Now, if you look at the functions of components, number one, generation of opsonins that you see, especially C3b. Number two, production of anaphylatoxin, we call it the A components. And number three, cell lysis that you see, membrane attack complex, MAC, which will polymerize and will create a channel and cause disequilibrium of water and ions. Now let's take these functions one by one. Production of opsonin, this is the most important function of complement, especially as a part of post defense. And it's generally considered to be the generation of fragments with opsonic activity that basically deposit on cell surface. There are two major opsonins which are generated, C3b and C4b. Remember, B is basically the portion, not an A, because A stands usually for anaphylatoxins. Now also keep in mind that IC3b does not activate complement, but it may have or does have an opsonic activity. So if you look at the major function of complement production of opsonin, especially you can see number top, opsonization. The opsonin will attach to the bacteria and they'll make them palatable to the phagocytes, the neutrophils, and macrophages, which will come and devour them or eat them. Number two, again, you will see production of anaphylatoxin. You'll ask yourself, why do we need to have anaphylatoxin? We need to have anaphylatoxin because of their ability to induce shock-like characteristics of systemic allergic or anaphylactic response. Now, in this case, if you pay attention to the middle figure over here, which says that there is a C5a, which is the most important anaphylatoxin, and then again there is a C3a. C4a is a little bit less important in terms, but all of them, as you can see from this figure, if you compare the vessel on the left to the vessel on the right, that they activate vascular endothelial cells. So by activation of vascular endothelial cell, they will increase the permeability of blood vessels. And then the blood vessel will allow the phagocytic cell to move in and out, will also allow bacteria to move in and out, and will also cause protein disequilibrium, and you may have a local edema there, especially in the tissues. So, of course, this will show you that anaphylatoxin, as the name suggests, are basically going to enhance inflammation and basically would cause the phagocytic cells to leave from the blood vessels to the tissues where there is a need for them to go and phagocytose bacteria. Now, the third part of it, as you saw, this is something that we already discussed, production of anaphylatoxin. I just explained that to you. Again, C5A is the most potent anaphylatoxin. You need to remember that. And then followed by C3A, C4A. Keep in mind that they all have A, and they are not opsonin. And then if you look at the third important function of complement function is that they will cause lysis of pathogens because we need to kill the pathogens. They are injurious to us. In this case, as we discussed earlier, without going in much detail, C439 polymerize and they form a membrane attack complex, which is called MAC, on the surface of the cell, and which will lead to osmotic disequilibrium with water and ion moving in and out and causing total disruption of cell membrane, and then the bacteria, of course, will die. Now, these were not only the only three functions that complement proteins have. We have a bunch of other functions there. And if you look at some of the functions that your book has mentioned, number one, we discussed earlier as well, they enhance B lymphocyte responses to antigen. And I'm going to discuss that in a while. Number two, they controlling the formation and clearance of immune complexes, important. Number three, removal of dead and dying cells. And number four, also keep in mind, it may have seemed to you that we discussed bacteria, but basically they also help us to get rid of viruses, and they also interact with viruses. Now, this was the slide that showed that complement basically enhance B cell response. In this case, you can see on the top is a enhancements of B cell response. So B cell comes, it has an antibody over there. It is going to look at antigen antibody complex, and FC portion of that particular antibody has a component which is there for complement fixation, C3DG, and likewise IC3B. Now, these are the two complement proteins that basically tie up to two important receptors expressed on the follicular dendritic cell. So you can see CR1 and CR2, and then basically it will bring these antigen antibody complexes together, and then they will be taken up by a follicular dendritic cell. So that was number one. Number two was in terms of B cell activation. You look at the B cell surface, and again, they are two different antibodies. They recognize that bacteria, and then there is another one which is a complement C3DJ, and basically it will latch onto the site of bacteria and communicate with the receptor expressed on the B cell surface, which is CR2, and that basically would enhance the signal, as we discussed earlier in B cell chapter as well, that this will kind of decrease the load. So you can basically look at a very low antigen load or very low bacterial load and still get a good immune response. So that basically will be a positive in terms of enhancing B cell function. Now, let's review them again. We saw the previous figure. There was binding of C3D or C3DG to CR2, and then of course enhancing antibody responses. And you saw especially C3DG binding to antigen, and then lowers the threshold for B cell activation. Normally, for example, if it's 1000, it's going to decrease that. So you basically would need 1000 bacteria to cause a particular effect, and then even one bacteria where it is bound to complement protein will have the same effect. So you will see that that's pretty much the function of C3DJ, which is enhancing B cell response. If you looked at the receptors which are expressed by the follicular dendritic cell, CR2, again, they will help, especially for antigen antibody components, so that they can be presented to the germinal center B cells, where you have a special B cells that can take that as a memory cell. So they will engulf that and keep them as the memory cell. So that again is an important, very important function when you're asked about the role of complement in terms of enhancing B cell responses to antigen. And remember, these are not the T cell, but the B cell response. Now, if you look at the role of complement in terms of getting rid of immune complexes. So you can see in the top figure, there are antigen antibody complexes. And then again, because agglutination, precipitation, all these different things. And then on the side of FC portion, there are places where there's a C3b attachment, complement fixation antibodies. So they'll attach over there, and then cells like, for example, macrophages and even red blood cells, they have CR1 or CR3 receptors, or even FC receptor. So basically, then they will be brought into the liver or spleen, and they will be cleared off. So you can see components, especially complement protein, will attach, because the danger is that antigen antibody complex can clog the whole system. They will go and deposit, as we discussed in allergy and hypersensitivity. They will cause many problems. So you want them to be removed and cleared off. So how do you do that? You attach C3b components with them and let these cells take them away to particular areas like liver and spleen, and then they are cleared off. The other important aspect of complement, you see, well, we have no infection. So what are these 30 complement proteins doing in our circulation? We need to remove those necrotic cells or subcellular membranes. So if they are cells, they have lived out their lifespan, they are necrotic. We want to get them out of the system. They are C3b and C4b. They are opsonin. Again, they will attach to it, tag them. Or if it is a dead cell, it has an old mitochondria. We want to get rid of that mitochondria. This is in not producing oxygen or whatever the function of that particular mitochondria is. So they will be again tagged by opsonin, and they will be presented to neutrophils and other phagocytes, like those having CR1 or C3 receptor, and they will be cleared off. So you can see that one of the functions, very important function, even in disease or in without a disease, is that they will clear off antigen antibody complexes, and they will remove necrotic cells and subcellular membranes or portions of some of the membranes which are there and need to be removed from our body. And finally, in one of the caption, I mentioned that we can also see the role of complement in terms of response to viruses. So there's a virus in this case, you can see from here. So you have C1 classical pathway, then you have lectin binding pathway, they're coming attached over here, and likewise you have C3 alternative pathways. So all these pathways will come and will help to get rid of that virus. And then again, as we discussed, classical pathway will have Ig, and they will basically go and take care of that virus, so that virus is removed from the system. Now, if you look in a broader picture of whatever we have discussed so far in terms of other functions of complement, and you can see on the top, we talked about enhancement of B cell responses, especially the follicular dendritic cell, and taking these to B cell areas in the cortex where you can keep them as memory B cells. Number two, you want to remove all those immune complexes from the circulation by the red blood cell and the macrophages. They're taken into liver and spleen, and they are cleared off. And then finally, we also wanted to find out if there is no infection, but there are dead or necrotic cells, or there are subcellular membranes like old mitochondria that needs to be removed again by phagocytes. They have those receptors for complement proteins, and they will be also cleared off. And finally, you can see all these three pathways will unite together and fight with the viruses that we have. So you can see from here that complement protein is a very important chapter, and this basically is important in antibody mediated getting rid of the functions. It also is important for initiation for inflammation. It's also important for getting rid of the immune antigen immune complexes, and they have 30 different plasma proteins. So I'll just repeat a couple of important things. I want you to know all three pathways. I want you to know how these pathways are activated, which pathways involve antibodies, which involve do not antibodies, what is MAC, a membrane attack complex, what is C3, what is different between anaphylatoxins, what is different between A and B, and then again, how would they help in terms of enhancing the B cell response, what is their role in terms of viral response, how do they get rid of necrotic cells, and how do they go about enhancing the B cell function. And one of the slides that I showed earlier as well, where we talked about especially a B cell related thing where this complement will decrease the threshold for B cell activation like 1000 times. So that's a very important concept that we need to know. And I think that pretty much will take care of whatever is there in terms of complement. And also, I did mention that some of the antibodies like IgM and IgG have sites on the site which are complement binding sites, and some of the antibodies have this ability to fix complements. And also keep in mind that the FC portion of the antibodies and where do these attack, and then also remember how do we regulate. So we talked about the regulatory factors in the serum which are circulating, and these are the factors will make sure that they are properly regulated and they don't run wild and cause all those problems.